Hello second year students, now we will try to understand that how can we prepare fungal slides. Okay, so first of all you can see very nicely that here in case of fungal media, this is rose bengal agar, you can see very nicely that fungi has grown in the form of colony. This is bigger colony which is of aspergillus and this is the another fungi. Here also in case of PDA you can see three different colonies. Okay, two are having white fluffy growth and another is having blackened growth. And this is another plate of rose bengal. Here also you can see different colonies. There are three colonies of black fungi and there are three colonies of the green fungi. And this is apex agar. In this also you can see different colonies which are merged into one another. Okay. So now how can we prepare the slide of the fungi? Okay, so that we will try to understand, and I will give you each and every thing very clear for your understanding. Okay, first of all you have to take one slide with you. Okay, and then after you have to take your pen. Okay. Now if you want to use this culture again, then you have to do this whole work aseptically, but because I have just want to demonstrate to you, I don't work in the flame area. So now I can open this plate and I will take a very little amount of fungal growth. Okay, now here you have to be very sure that you are going to touch the white part as well as the black part. So white part includes somatic structure and black part includes the sporulated structures. Okay, and then after you have to take the staining solution, that is called lactophenol cotton blue dye or lactophenol solution. Now in this particular solution, because it is very darker in the color, we are trying to you can say dilute it out with the help of distilled water. So we are able to see the parts of the fungi very nicely in the microscope. Now I have taken the fungal material in the tip of this pin, and now I will try to disintegrate this particular material with the help of these two pins as much as possible. Okay, I will carry out this particular thing for 5 minutes. Okay, this is the work of patience. If you are in a hurry, you cannot give the good result. Okay, so properly try to separate the mycelia of the fungi, and at the end of that you have to take one cover slip with you. Okay, now you have to hold this cover slip at 45-degree angle with the help of this pin on the glass slide, and now immediately with all precautions that there should not be any bubble in the slide. So in between cover slip and slide there should not be any single bubble, and gradually now you have to immediately drop this cover slip on the slide. Okay, just you see like this. Okay, and now you have to do this for 3 times, 1, 2 and 3. Now your slide is ready. With the help of filter paper you can try to absorb all the material which is found outside the cover slip. Your slide is ready now. Now this slide can be observed in the microscope using low power, then after if it is required then in high power also. Okay, so this is about the fungal mounting. Okay, now suppose your fungi is very velvety. Okay, then how to prepare that type of fungal slide? So first of all we will take the slide and we will take the very small drop of this lactophenol cotton blue, and we will dilute it out with the water a little. Okay, so now we have diluted out because it is very dark and so we need to dilute it, otherwise not. Then take 2 fresh sterile pins, and with the help of that, okay, you can take the material of the colony which is appearing velvety. Now here also you have to be very sure that you are going to get the green growth as well as the growth which is white in color. So all the parts will be there, somatic structure will be white and green coloration will be of the spores. Now you can see that very nicely that our fungal structure is there. Okay, it is very obvious. Now you have to disintegrate this granular material with the help of the head of the pin. Okay, now this thing you have to do for so many times. Not only once you will get the result, but for 5 minutes you have to dig this out. Okay, very gradually. Okay, with all the patience and for 5 minutes like this. Okay, and if this is very difficult for you, you can take the another slide also, and with the help of the one side of the slide also you can disintegrate like this. Okay, so this will also give you good separation this way. Okay, fine. So you have to be very patient. We are trying to separate out the granular part of the fungi. Now we have already done the slide. Now what we will do the next, that we are going to apply cover slip on the glass slide. Okay, so for that reason we will take the cover slip now in our hand. Okay, now I will just hold this cover slip with the help of the pin. Okay, and then gradually I am just putting it down with all precautions. There should not be any air bubble. Okay, so now our slide is ready. You can mount this in 10x and also 45x, and you can observe the fungal structures. Okay, so this is the way how we are making the fungal slides. We are mounting the slides. You can mount all these structures which are already there in the form of colony. Okay, and this is really interesting and very easy to do, and you are going to get very beautiful spore structure, reproductive parts of these fungi. It is really very good to know this beautiful color fungi. Okay, thank you so much.